Alright guys, so my last video left off here where I was talking about the cell during anaphase. So during anaphase, the nucleus has now completely disappeared. The centrioles are at either side of the cell, and the spindle fibers are going to be attached to the kinetochores. So that's that little protein piece that holds the two sister chromatids together. So the spindle fibers will attach to the kinetochore, which is located at the center, and then the spindle fibers have started to pull the sister chromatids apart from each other. And this one is very, very, very easy to identify when you're trying to classify the cells. I'm sure you guys figured that out from the little virtual simulation type lab that I had you guys do on Tuesday. So if we look at our picture, remember, like I just said, it is the sister chromatids that are being pulled apart from each other. And once they do get pulled apart from each other, we can then call them chromosomes again. So what ends up happening is we're going to get two new cells that have 23 chromosomes from mom and 23 from dad, all these blue ones, for a total of 46 chromosomes. The other key that you need to remember about anaphase is that not only are the spindle fibers attaching to the kinetochores to pull the chromatids apart, to pull the chromosomes apart, but also the spindle fibers don't all attach to a kinetochore. Sometimes the spindle fibers end up just like hitting each other. And so the job of those, and what you see during anaphase, is the cell goes from being a circle in metaphase to kind of becoming elongated like an egg, like an oval, whenever those spindle fibers start attaching to the kinetochores and then also to each other. So basically, if my fingers are representing the spindle fibers, some of them will hit each other, right? And what they'll do is they'll push and they'll push and they'll push. And so they're pushing against each other, which is pushing the cell apart. So I don't know how many of you have siblings, or if you've like done this with your friends. Me and my best friend used to do it all the time when I was little. But like you and your friends sit on either side of the couch, and then you put your feet together, and then you push against each other's feet, and whoever like bends their knees up to their chest first loses, and whoever is able to like straighten out their legs wins. That's kind of what's going on with the spindle fibers that don't connect to the kinetochores, but they connect to each other. They're just kind of playing that game of like the foot war. I don't even know. It didn't even have a name. We would just do it. But when they push against each other, it's like you pushing your feet against your little sister's feet, or against your friend's feet, and you're pushing, 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 which is like separating you guys. Obviously, you have the arm of the couch holding you onto the couch, but if the arm of the couch wasn't there, you'd be pushing each other off the couch, basically. And so that's pretty much what's going on with the spindle fibers. So in anaphase, the cell is no longer a circle. It's more like an oval or an egg shape. And then lastly, we have telophase. I've kind of just drawn telophase and cytokinesis really as just like one thing for the sake of saving paper. It was kind of running out of space. But the last step after anaphase is telophase and cytokinesis. At this point, the nucleus has started to reform. It's almost completely formed. By the end of cytokinesis, it is completely formed. But in telophase, the nucleus starts to reform. And you can see that you have 23 chromosomes from mom and 23 chromosomes from dad for a total of 46. And over here, same thing. Now, the reason why over here I kind of drew the red and the blue all mixed together is because that's technically what would really happen. They wouldn't stay perfectly separated like they're staying over here. They'd really be more like mixed in with each other. And again, they really don't even have a color. But Again, I just drew them as colors because it will help us whenever we start meiosis, which will be our next thing. So if we look at these cells down here, we'll notice that they look exactly like the picture that we started with. And that is the goal. It is to get 2 new cells that are genetically identical to the parent cell. So the daughter cells must be identical. If those daughter cells are not identical, then that means there was some issue that happened in one of the phases, and maybe there wasn't like an appropriate check, or somehow the cell managed to get past a checkpoint that it shouldn't have. Again, that does happen. That's when we get things like cancer. Cancer is, like I said in the last video, cancer has this amazing ability to just turn off all the checkpoints that are there to make sure, okay, yes, you passed this, you have a copy of all of your chromosomes, good, this is how it's supposed to be, go forward. Oh, okay, you've all lined up on the metaphase plate, good, you can go to the next step. For some reason, cancer cells are able to just turn those off, and they just start replicating and making copies of themselves and undergoing mitosis, even when they shouldn't, and even when stuff is wrong. But for the most part, normal cells, most cells, follow this process. So I hope hearing it from me has kind of helped you to piece together all of the things that you learned in the articles that I had you guys read and in the videos that I had you guys watch. So thanks for tuning in with me. Miss you guys.